Okay, we'll get started. Can everyone hear me okay at the back? Okay, great. So hi everyone, I've been sort of hanging around in the background here, but in the shadows. But my name is Shweta and I'm a faculty member at the Center for Brain Research, which is sort of off, right, on the opposite side of the road as ISC. We do a lot of sequencing studies. So one of our flagship projects is this consortium called Genome India, where we're trying to sample genetic variation from around the country and trying to really understand the genetic diversity of Indian populations. I'll say this again at the end, but extra advertising never hurts. So I'm always looking for students and postdocs. So if any of you is graduating, looking to move into human genetics, please come talk to me. Yeah. Okay, so for the next hour or so, we'll talk about genome-wide association studies, or as we call them, GWAS. This is slightly off-center, in the sort of context of this workshop. Kavita had mentioned there's population genetics and there's quantitative genetics, right, 2 days ago. Genome-wide association studies fall fairly squarely into the realm of quantitative genetics or statistical genetics, right? So in this next 1 hour, I won't be explicitly talking about, a lot of the principles that Kavita and Deepa have spoken about, but they're implicit in the experimental design of the study. So one thing that might be fun is for you to connect everything I'm saying with the underlying principles of evolution and popgen that are sort of going on in the background, yeah? Okay, but before I start with GWAS, I thought I'd show you a couple of sort of vignettes about human genetic variation, since that's what I work on. So, okay, so let's look at this result from the 1000 Genomes project. This is this gigantic project that tried to sample or sequence the first 1000 human genomes, okay? They did that, and they plotted this plot in their paper where they plotted the number of variant sites per genome across different human populations, right? So the different colors represent different major human populations. In shades of yellow are populations from Africa. In shades of red are South American populations. These are South Asians, East Asians, Europeans, okay? Okay, so right off the bat, I'm going to ask you to interpret this for me. What do you think would explain this? There is an underlying historical process or underlying population genetic event that has led to a pattern, to the sort of most striking result of this plot. What can explain the most striking result of this plot? What do you think? What do you know about human history that you think could explain this? Migration, what do you mean? Can you first tell me what it is that is most striking to you, and then what you think could perhaps explain it? Okay, so maybe I'll explain the y-axis a little bit better. What the y-axis says is, if you take a single genome, how many sites on this genome show a difference from the reference allele? Okay, so what this says is that African populations seem to show the most number of variant positions in the genome compared to every other, by a considerable difference compared to other populations. So do you still think it's migration? Okay, yeah, think about it. Yeah. Yeah, go ahead. These could be polymorphic. Yeah, these are actually polymorphic, right? Yeah. The reference genome is this very imperfect reference genome it's a mishmash of 3 or 4 people, most of whom are European, but there's one African American individual in that reference genome. Yeah. Huh? No, it's actually 4 or 5 individuals who are, they sequence them, and then we don't quite know the ratios of populations used to create the reference, which is strange given we use a reference for everything, but. Are they? To create the reference? They've done it, but I don't know about assembly. I don't know if I know Craig Venter's genome has been assembled from scratch, but that's still not in the reference yet. The reference is still this mishmash. Yeah. Can you be a little louder?
Your reference mostly comprised of Europeans.
Yeah, so some of this is likely technical, right, where the reference was likely European, but there is a biological event there is a biological process related to something that Kavita has spoken about before that could explain the higher number of, maybe related, I don't know if you spoke about it yet, but something about human history, yeah. One sec, huh? Go ahead. Yeah. Yeah, exactly. That's really it, right? I don't if all of you didn't hear him. He said it was the founder effect, and that's exactly what explains that. It's that human populations originated in Africa, and so most human genetic diversity sort of was in that original huge population. A very small number of individuals then left Africa in different waves of migration, right? So one wave sort of followed the South Asian, the southern route. The second wave sort of taking the northern route. Essentially, all non-Africans represent a bottlenecked population, a much smaller fraction of the genetic diversity in Africa, which really explains why most human genetic variation is contained in African populations. Um, yeah. 5 million variant sites per genome. Yeah. Okay. This was also, this was also 8 years ago, so the numbers are much larger now. But qualitatively, this stays the same. Okay, so that was sort of just a little bit of popgen before we move on to GWAS. Okay, actually before then, another thing I find really useful when thinking about population genetics and genomics is to develop an intuition for numbers, right? Like what are the orders of magnitude we're thinking about? And so maybe I thought we would just think about these and then I'll give you the answer. So what is the length of the human genome? 3 billion, yeah, so 3 into 10 to the power 9. Okay, so the human genome is 3 billion base pairs long. How many mutations do you think arise per generation? Let's say, right, so mom, dad, your genome, you have inherited 3 billion base pairs from mom and dad. How many of those do you think have mutations not seen in either parent? Just to give you a sense to get you thinking about these orders of magnitude, right? Do you think it's 0? Do you think it's 100? Do you think it's 1,000? Where do you think the number of new mutations lies out of 3 billion? Out of 3 into 10 to the power of 9, how many mutations just arise when you're in the, yeah? 100, 200, yeah, so the answer is close to 60, right? So the mutation rate is about once every 10 to the power of 8 bases. So yeah, about 60 new mutations. Each of you carry 60 new mutations that came from neither of your parents, right? Okay, so what is between any two humans, what is the average genetic difference? Out of 3 billion bases, if I take you, take somebody else randomly, compare your genomes, how many are going to be different? How many out of the 3 billion are likely to be 1%? If it were 1%, one of you would be a chimpanzee. Huh? Greater than 0 but less than 0.5%. Yeah, it's 0.1%, right? So it's 1 every 1,000 bases on average differs between two individuals. Yeah. Okay. Yeah, and another one. How many human genomes have been sequenced until now? Guess. Again, you don't know these answers, so you can just guess because you could be wrong and it'll be okay. Many? Yeah. Yes, many. Well, okay, I'll tell you the answer to this because this is a fact that makes geneticists who work on other model organisms really hate human geneticists, right? Because your favorite species has not even been sequenced and we have like a few million sequences just lying around for anyone to study, right? So a few million maybe, 1 or 2 million genomes have probably been sequenced until now. Whole genomes, right? Which is like no other species even comes close, right? And how many genes? So all of this is another advertisement to come work with me because human genetics data, we have a lot of data to work with and we know very little. Okay, and finally, how many genes do humans have? Yeah, depending on how you define gene, between 20 and 30,000, right? Okay, so this was just to really get you a sense for some numbers that human geneticists sort of regularly think about. We spoke about this. Okay, but what is a genome-wide association study? The sort of core question that a genome-wide association study tries to ask or answer is, how do you find the genes responsible for a disease? And more specifically, how do we find the genes responsible for a disease using data from human populations? Okay, so this is what a genome-wide association study is. There are variants of this in different organisms like mice or rats, but I'll sort of focus on how these are done in humans right now, okay? If you might have heard of these companies like 23andMe, where you spit into a tube, send your DNA to them, and they will sort of return something that might look like this, right? So this is from somebody I know where they give you, this person had a high risk of these disorders, and what they return is your particular genetic risk for a disease compared to the population risk, right? So what we'll try to get at today is, how do these studies help us arrive at this, at something like this? Okay. Okay, so human genetics didn't really start out with GWAS. Human genetics started out with pedigrees. We geneticists love these, right? We started out looking at genetics as disease, this as being inherited in families. So in the 70s, before sequencing was a thing, what we would do, what geneticists would do is work with clinicians, work with doctors, go and look at pedigrees where you saw a high inheritance or a high prevalence of a particular trait, right? A particular trait of interest. Go and try and sequence the individuals carrying the trait to figure out what parts of the genome were shared between cases, right? This, however, worked very nicely for Mendelian diseases, right? This worked for a whole bunch of Mendelian diseases. We could find those genes, but very soon we started finding that the genes that were responsible for one disorder in one pedigree could not be replicated in other studies or in other pedigrees. And that's because, as I think Kavita had mentioned earlier, most traits are not Mendelian but complex, right? So most traits are not governed by one gene, one disease, but by many genes plus the environment together sort of influencing your risk for a particular disease. What this means is that these traits don't have to run in families. If your mom or your grandmom has a disease, you don't have to have it. It also means that different people with the same disease trait may have different factors. Yeah. So for some diseases, the environment may not matter. Like for phenylketonuria, it is a metabolic, inborn error of metabolism, so you have it irrespective of environment. A lot of them are severe enough to show up irrespective of, essentially to show up because the environment in which they wouldn't show up is not really studied, if that makes sense. Can you tell me more about what you mean? Yeah.
I thought maybe environment should matter for any number of, there's a Mendelian complex.
Yeah, yeah.
So, but I always see this discussion which they talk about Mendelian, but environment is somehow not in the picture, and I don't understand it like that.
If you imagine a mutation in a disease gene that's at the sort of core of a pathway, it means irrespective of the environment, the absence of this gene function is going to affect phenotype. Yeah.
Why not for Mendelian?
With complex traits, I mean, I was going to later talk about a lot of these are sort of driven by a lot of smaller effects. And therefore, whereas Mendelian diseases, the effect sizes are huge. I mean, I think, okay, so you're not wrong in that even when we talk about Mendelian diseases, a lot of the variants now that we have associated with Mendelian diseases are incompletely penetrant. Just because you have the variant, 99% of the time you will have the disease, 1% of the time you will not. And we usually don't know what happens in that 1% of the time. And you're right, maybe it's sort of environment, maybe it's, yeah.
You said the environment will matter, but it's negligible.
It's yeah. Okay. Okay, so if you look at something like height, you might think that height is influenced by multiple genes, but also environment, your diet, life events, high stress, I don't know, but also interactions between genes and the environment, right? Certain genes might, the effects of certain genes might show up only in certain environments. And most diseases, most traits that human genetics study are sort of fall under this paradigm of being affected by multiple genes, environment, and interactions between these two. So again, going back to the paradigm of a GWAS, these are studies aiming to find genetic differences between individuals that influence susceptibility to diseases. And here, note that I'm no longer saying cause diseases, right? Because it's no longer a single variant causes the disease. It's it's variants that increase your risk for having a certain disease, right? So that's what GWAS try to find. Okay, so the core principle of these studies is really very simple, very intuitive. You collect a large group of people. Let's say, pick your favorite disease, right? Pick your disease that you really care about. Let's say diabetes, right? A group of people who have it, a group of people that don't, sample their DNA, and look for genetic differences between these two groups of people, right? It's quite simple conceptually, yeah? Okay, so this is what we'll talk about, but okay, so let's talk about that second part, which is look for genetic differences between the two groups. How exactly do we do that in the context of a GWAS? How do you think we would do that? We would see. Okay, so how would you find the number of single nucleotide polymorphisms? Okay. Okay. Yeah. Right. Yeah. So you said, the way you find these genetic differences is by sequencing all of those two groups of people, right? But let's go back to variation. I'll come back to what you said, right? So we know the human genome is 3 billion bases long. What we're going to define is that the positions we're going to test for association with the phenotype are going to be called single nucleotide polymorphisms, or Snips, right? These are positions that sort of are commonly varying in populations of individuals. They usually have 2 alleles, C and G, just to make sure everyone is on the same page with these definitions, okay? We don't actually sequence all of these individuals in a GWAS. We'd like to, but sequencing is really expensive, right? So right now, sequencing a single genome would cost you more than 1 lakh, right? And imagine you want to run a GWAS with tens of thousands of samples. We're going to run out of funds very quickly, right? So we can't really sequence every position in the genome. We would like to test all of the 3 billion positions in the genome to associate with the disease. But instead, we need to sort of be smarter about it. Can anyone guess how else you would do this? If you were faced with this problem, right? How would you do it? You'd like to test all 3 billion positions of the genome, test if these are associated with the disease, but you don't have the funds for it. So how might you think of a smarter way to do it? Or a cheaper way to do it? Sorry? Reduce what? What is, so reduce representation sequencing? Okay, so what is that? Okay, so I think both of you are getting at the same thing, which is maybe don't sequence the whole genome, only sequence the protein coding part of the genome. Something like that, where you're only looking at the genes. That's one way people do that. But genome-wide association studies use something else, which is they use, again, going back to linkage, they use a property of the genome that comes from linkage, right? So we all know linkage, where we all know about recombination, where let's say these are the mom's chromosomes, dad's chromosomes. We inherit a mosaic of chromosomes, but because of the relationship between recombination and distance, A and B are not going to be independent of each other, right? So your genotype at A is going to be dependent on what your genotype at B is, right? So if I know that my genotype at A is something, I have some information about what my genotype at position B is going to be, right? Not true for A and C. A and C are likely, they're far away on the chromosome, so they're more likely to be independent, right? So what this means is that positions that are close together on the genome travel together, and we use this property to sort of test only a subset of positions on the genome, and these are called tag SNPs. So what we do is we've defined a set of variants in the genome. So this is a single chromosome that almost stand as a marker or a flag post for the region around the SNP, right? Because if you know this variant, the positions around this variant are likely to travel together because of there's less likely to be recombination in this region, and therefore this marker stands as a sort of proxy for this region of the genome. Okay? So instead of sequencing 3 billion positions in the genome now, we're only genotyping 1 million, right? And this is vastly cheaper. Yeah. Yeah, mutations can happen, but that's why we're sort of going back to what genome-wide association, the kinds of variants they test. They look for single nucleotide polymorphisms, right, which are common. So this kind of study only looks for common variants. It does not help you find rare variants. Yeah. Okay. Okay. So this is how you run a study. Let's say again, pick your favorite disease. You collect cases, collect controls, get DNA from each sample, fragment this DNA into pieces, and because now we already know what positions we want to genotype, these 1 million positions or so, they have probes for each of these 1 million positions on, yeah.
So which one?
Yeah.
So you're saying that we do 1 million tags. So what exactly? The box part is the type, or
We only genotype these positions. Yeah, so a very small fraction of a chromosome is actually genotyped on a chip. So, yeah, exactly. Yeah, exactly. Exactly. Yeah, because from that first 1,000 genomes project, we know that these tag SNPs have been selected in such a way that the region around them is in high LD. We know that recombination occurs a lot less frequently in this region, and therefore we pick this marker as like a flag. Okay, so we collect DNA, and the way we genotype them is not using sequencing, which is again still very expensive, but using a chip where each of these 1 million positions has a probe, right? This probe lights up differently if you have allele A versus allele B. Okay, and so what you get from the data you get from these studies is a matrix of all your subjects, cases, controls, and all your M SNPs. So this would be 1 million by how many ever samples you study. Yeah. Within this matrix, let's say in row 1, you can have any of 3 possible genotypes, right? 0, 1, or 2. Where 0 is 2 copies of the reference allele, a hetero is 1 copy of the reference allele, 2 copies of the alternative allele, right? Okay, then what we want to do is actually run the actual tests. So each of those 1 million SNPs, we want to see if it's associated with the disease, right? So what you want to see is something like this, right? Where in cases, most people have an A. In controls, most people have a T. Let's say the 2 alleles are A and T. Okay, does that make sense, right? This is the kind of thing we want to see, right? If we see something like this, we say, aha, having a T makes you way more likely to have the disease, or the other way around. So that's what you test. We test the significance of association of the 2 alleles at this particular SNP with the phenotype, and we use what's called a chi-square test for it. And this test gives you a p-value. All of you familiar with p-values? Or anyone not familiar with p-values? Okay. Yeah. These are 6, let's say, it's a cartoon of haploid individuals, so slightly not representative. Yeah, 6 individuals. These are cases, these are controls. And what you see are cases mostly seem to have A, controls mostly seem to have the other allele, and therefore you think having a T makes you more or having an A makes you more likely to be a case. Yeah. You want to see something like this. And so you test for this kind of pattern across all million positions, yeah? And each of these tests gives you a p-value. We love 0.05, so if it's less than 0.05, we call it significant. Do you agree with that? Any of you, I know some of you are from math backgrounds. Is there a problem? Or if I ran these tests gave you a list of all variants that were significant at p-value less than 0.05, published it, would this get into Nature? Huh? We will look at the effect size, okay. But are those, let's say I find 10,000 variants that are significant at p-value less than 0.05, 10,000 out of 1 million. So are all of those 10,000 really interesting? Huh? Not always, okay. Okay, what I'm trying to get at is that, oops, I'm going to come to this later, but what I'm trying to get at is the p-value threshold for a GWAS is not 0.05. We are running 1 million tests, right? We're not just running a single statistical test. This is a problem called multiple testing in statistics, where if you test the same thing like an infinite number of times, just by chance, many times you're going to hit your p-value significance threshold. Right? And that's called multiple testing. And in GWAS, it's not trivial, right? You're running 1 million tests. You're not just running 2 or 3 tests. So you cannot use a p-value threshold of 0.05. What you use is a much more stringent threshold, which is 0.05 divided by the number of tests you run. So we only pick an associated SNP is one that has a p-value of less than 5 times 10 raised to minus 8. Yeah? It's, I guess the contemporary analogy to this is, if you take 10,000 selfies and one of them turns out to be really good, it doesn't mean that you're a good photographer. It doesn't mean that I'm really good looking. It just means that you've taken 10,000 selfies and, one of them turns out to be really pretty and really nice and Instagram worthy. Okay. Okay, everyone okay with multiple testing? Okay, so I'll just go back. So the chi-square test sort of was for a binary trait, right? But what if you're testing something like height? You can also use GWAS to test quantitative traits, height, cholesterol levels, pick a trait that's continuous. In that case, these are the kinds of models you use, a linear model, where what you're testing is, you can have 3 different genotypes at each SNP, 0, 1, or 2. Do you see a linear association between individuals, or between the genotype and your phenotype, whatever that phenotype is, right? Height, insulin levels, whatever, okay? For those of you who are mathematically inclined, what we use is a very simple linear regression model where X is your genotype, you're regressing your phenotype, Y, on X. Beta here is what's called the effect size, right? It's called how much does one additional allele, how much does it make you taller by? Let's say if height is your phenotype. Or how much does it increase your cholesterol levels by? That's called the effect size, and that's what's represented by beta 1, which is, in this case, the slope of the model, right? The slope of this model is the effect size, and the other quantity of interest is the p-value, okay? Yeah. Yeah. Correct. Yeah. Yeah. This is measured. Yeah, this is measured data, right? So this is the height. We fit a model separately, and we can kind of do that because we're going back to that tag SNP bit, right? These are, because we've selected them to be fairly independent, we can test an independent model for each of these loci separately. In practice, we test them separately for each SNP. Is that what you mean? What do you mean by locus? Yeah. Yes. By locus, do you mean SNP? Yes. Yeah. Exactly. And that's why we need to correct our p-values, our p-value threshold, right? Yeah. Yeah. Yes. Yeah. So the effect size is actually I might have a separate slide for that. Okay, yeah, here, this is what the effect size is, right? It's not, this is just to sort of tell you, it's not, we speak about p-values a lot, and p-values are great to help us sort of pinpoint SNPs that are important, but from, let's say you're a biologist, what you care about is how much does your genotype influence your phenotype, right? And for that, the effect sizes is perhaps a more useful metric, where all of these 3 different examples, three different Snips have a similar p-value, but the effect sizes are very different, right? Where you can see that having the T allele in this case increases your height maybe twofold. In this case, it increases your height by fivefold. In this case, increases your height by sevenfold or tenfold. So that's what your effect size is.
So beta naught is some shift.
Yeah, beta naught is the height, average height for the, yeah, error. Environment, error. We are clumping so that is the deviation from the linear model. E is, no, beta naught is the intercept. E is everything that cannot be explained by the model that we assume is independent of the genotype. No, E is just the error term in the linear regression model. It's
for each point there.
E is not something that you put into the model. E is something that that comes out of the model, right? No model is perfect. What you put, okay, so let me make that clear. Sorry, let's revise, let's go back to linear models again, okay? What is the input to the model? Two columns. One is what is your genotype, what is your phenotype? That is the input to the model. What the model finds out is what is this slope. Essentially, how much does this increase, right? And what your model also tells you is, okay, what is the average height for a person having this allele? That's beta 0. And E is how much can the model not explain, right? It's not like all of individuals with genotype 0 have the same value of the phenotype. Whatever the model cannot explain is sort of dumped into that E variable, right? It's noise, it's error. Does that, Kavita, did that explain? Exactly, you're trying to minimize the error, yeah. It's a usual error, but we're also collapsing the environment into error in this case, yeah. Yeah, okay, yeah. So, I mean, one way to think about, biologically to think about nonlinear models could be dominance or recessiveness, right? But, and I mean, sort of the biological underpinning of this is it's an additive model. Having one extra risk allele will increase your phenotype a little bit. Having two copies of an allele will increase your phenotype by twice that amount. In human genetics, most loci seem to follow the additive model really well so far. So dominance is actually a lot more uncommon, so we don't test that very often. Nonlinear models, I have not seen them being used. Outside of, you mean outside of dominance, recessiveness, additive? What other kinds of nonlinear models we've not really tested? No, yeah.
Do you know which other organisms people have their nonlinear models? Like plants maybe?
I'm not aware. Do you know if plant they use nonlinear models? Okay. Yeah. 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 Exactly. Yeah. 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 Okay. Yeah. Yeah. No. Yeah, sorry, I wasn't clear about this. No. A chi-square test is done if your trait is binary. You use a linear model if your trait is continuous. Right? So if you're working with height, you would make 1 million linear models. If you're working with diabetes, status or not, you would run 1 million chi-square tests. Yeah? Yeah. Every individual would, no. So let's do a cartoon one. No, I'm glad you brought this up because I have not been clear. So let's do this. Okay. Okay. So row 1 is genotype AA. Okay. I'm assuming all of you have the same genotype, have been selected for having genotype AA at SNP 1. Row 2 has been selected for having genotype AT, and row 3 has genotype TT. Okay. All of you in the same row have one genotype. Okay. And all of you have a variation in heights, right? So let's say your height is here, your height is here. I'm going to plot 1, 2, 3, 4, 5, 6. I'm going to plot 7 points here. Okay, now row 2, all of you have a different maybe a different distribution of heights. All of you with genotype TT, maybe your heights look like this. Okay. The best model that fits this is maybe aligned like this, because there is really no association between this imaginary genotype and this imagined height. Okay. If the 3 rows had 3 very different distributions of heights, let's say one of you in row 3 was this tall, one of you in row 1 was this short, then maybe there'd be a model like this, and that would be a variant that would be significant. Okay, does that make sense? Yeah. 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 So the question was, how do you know the beginning of a GWAS? How do you know how many samples to start with, right? How do you know if you start with 100 samples, 1,000 samples, 10,000 samples? And that's really hard, because the power you need to detect a certain variant depends on what the effect size is. If we expect our variant to have very high effects on the phenotype, then we can find it even with a small sample size. But if we expect our effects to be very small, we need more and more samples to find it. And yes, yes, yes. I'll show. Yes, that is very. I mean, you sort of hit the nail on the head. The number of samples have to be very large. Current GWAS sizes are, we're reaching 1 million or so. What do you mean by threshold power? We're testing it. We're setting the threshold for significance of false positive rate of 5%, right? We're setting that as our threshold, right? P-value of 0.05. Yeah. Okay. Yeah. We do, right? So let's say we have diabetes. How do we calculate effect size? For that, we would have something like this, right? Where you have cases, controls, variant A, variant T. What they're the effect sizes, essentially the product of these two divided by the product of, it's called the odds ratio, right? How much more are you likely to have the disease if you have the risk allele compared to the non-risk allele, right? So the term we use for a binary trait is the odds ratio. Okay. Okay. The top. Oops. Okay. So the bottom line here sort of talks about additional covariates that you feed into the model, right? We don't just expect height to be affected by genotype. We're working with humans here. The advantage of working with humans is that you have 1 million genomes to work with. The disadvantage is you cannot control for anything, right? You're working with what you get. So what these studies also try to do is control for other environmental factors that could affect your phenotype, right? So it's not just regressing your phenotype against genotype, but also trying to take into account anything else that you think might affect the phenotype and can be measured. That's a big thing. Whatever you think can be measured. There's always a possibility with human genetic studies that you haven't measured something of interest. Okay? Okay. So let's go to where we started with. We started with this 23andMe result, where for colorectal cancer we saw that my risk was 8.9% compared to a population risk of 5.6%. So how was that calculated? That was calculated because somebody had run a GWAS on colorectal cancer, found one SNP with a p-value less than 5 times 10 raised to minus 8. That SNP had an effect size of 1.6. And I had that risk allele, so my risk was now 1.6-fold. Okay, so you run the GWAS on one sample. Based on that, you can use to predict the genetic risk of another sample. Okay, yeah. The effect size from the model, right, or from the, yes, yeah, that's the effect size, yeah. Okay, quick, we have 15 minutes. How many of you have I lost completely? please raise your hand because I luckily have one more lecture tomorrow, so we can definitely sort of go over this again if it's not clear. How many of you have I lost completely? No judgments here. Okay, everyone with me? Yeah. Yeah. So, yeah, so effect size is what you get from your GWAS, from your model, right? The odds ratio or the effects of that linear regression slope. 5.6 times 1.6, essentially 5.6 plus like 1.6 of that is my risk. I have an added risk of 1.6x. Okay? Yeah. Yes. Yeah. Okay. Yeah. Yeah.
Are those positions targeted or they are randomly?
They are not targeted for function. So it's not like you pick these SNPs based on, okay, I think these genes are going to be associated with height, so I will pick these SNPs in these genes. So they're not biologically targeted, but they are targeted in that what do we want when we do use tag SNPs? We don't want to sequence the whole genome because we don't have the money. So we want to genotype as few positions as possible that have as much information about the genotype as possible, right? So they're chosen to be the maximally informative about the entire genome.
So they are one in a way targeted. That means the positions that are
Yes,
tagged are targeted.
they are targeted, yeah.
So then if we are sampling 10,000 individuals
Yeah,
and there are 1 million tags,
yeah, yeah.
and after tagging, those positions are being sequenced, right? So to find out those positions, one has to run through the whole genome, scan the whole genome. Or, so I don't see how the cost is being reduced.
I see. I will go through the trip again. No, you're right. I'm glad you brought that up so I can clarify it, right? Sort of again, we're not actually doing any sequencing here. We're not using next generation sequencing, right? Yeah, please ask me whatever is not clear, right? Yeah, none of this is, you're not expected to know any of this. So please ask me even if you think everybody else knows. Yeah. Every individual has to have? Yes, every individual has one. I mean, these chips can, sometimes have 96, but yeah, in this case, every individual has a chip, right? Yeah. 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 Yes. Yes. Yeah. If it were arbitrary, we wouldn't know where it would come from, where in the genome it would come from, right? Now we know that SNP A comes from position chromosome 1, position 500. Right? Yeah. Sorry, was that your question or were you? Go ahead. Yeah, go ahead. Yes. I mean, we don't know. We're testing for association. We're testing as many. This is still a hypothesis-free test in that we still want to find, again, going back to our goal, we want to find positions in the genome associated with a disease. We have no idea what these positions are. We unfortunately don't have the money to sequence every single position in the genome. So we're trying to be sort of hypothesis-free while also reducing the cost to run such a test. So we're picking at random, but not really at random, positions from the genome and testing only those positions. These positions capture information about the genome around them. So the amount of information we get is not just about these million positions, but about a greater section of the genome. Yeah? Yeah. They're not perfect. No technology is perfect, but they're, in terms of error rate, is that what you're getting at? They're very good. They're in fact much more reliable, or at least sequencing I think has a 95 to 99% error rate, right? This is actually better. So maybe the error rate is even slightly better than sequencing. So they're at least 99 to 99.5% accurate. Yeah. You can calculate the marker density, right? 3 billion positions in the genome, 1 million are being tagged. So you can calculate the marker density, right? Does that make sense? Is that what you were asking or what? 1 million. Yeah. It's between 500,000 to 1 million, again, within, depending on what chip you used, what kind of population you're working with, so on and so forth. Between 500,000 and 1 million is yeah. 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 Oh yeah, for humans, yes. Everything I'm saying is, yeah, with humans, yeah. 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 Yes. Yes. So again, going back to how I think the sticking point is what are these tag snips that we've picked? Those tag snips have, again, use the term tag snip. They're tagging something. They're tagging variation because they were found as being commonly polymorphic in the first set of genomes that were sequenced. Okay, so they're known to be polymorphic previously. They're known to very nicely tag the region around them. The region around them has low recombination around them, and yes, so therefore they must be common. Yeah, so tag snips are by default common. They're not rare mutations that you might see in one individual. Yeah. 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 That's a good idea. Okay, so let's do it for. Okay, let's do a cartoon example for height now. And tomorrow when I come back, let's do the actual thing for colorectal cancer where I can go and look up the variant. Yeah. Okay, so let's say I do the same. I use all of you to do my GWAS and height. Okay, so there's 40 of you in this room. I'm testing, let's say, one variant with height. Okay, so those of you with genotype AA, which I'll call the reference genotype 0, have this distribution of heights. Those of you with AT have this distribution of heights. Those of you with TT seem to be a little taller than average, and you have this distribution of heights. Can everyone see the board here? Okay, great. Okay. So we feed this into the model. We build a model from this data, from these data. This is height, right, on the y-axis. Okay. This is what this is the best line that fits the data. The slope or the effect size is something essentially y2 minus y1 by x2 minus x1. So let's say this is 1.1, which means that for every additional t allele that you have, your height increases by 0.1 centimeters or 0.1 millimeters, depending on what your height is defined as. Okay. Okay. So that's what your beta is equal to 1.1. Okay. Now, I am this new person. You want to predict my height. Okay. And my genotype is TT, which means I have two risk alleles or two height increasing alleles. So my genotype is going to be the baseline genotype of the population, which is the baseline genotype of anyone with the non-risk allele. Let's say this is 5 feet or 160 centimeters. Yeah? My units are all wrong. What are the units of 160? Anyway. Feet, right? No, centimeters. See, my units are. I started this lecture talking about developing an intuition for numbers, and here I am. Okay, okay, 160 centimeters. And I have two risk height increasing alleles. So therefore, my height would be 160 plus 1.1 times 2, which is 160. Okay. Yeah. If we find more than one variant, let's say at another variant, okay. I genotype again all of you at another variant, divide you into 3 groups, and at this variant, having this allele reduces your height. So your beta in this case is negative, is minus 1.1. okay? And so my genotype at this variant is also this DT. Okay? So what would my height be then? What would my height ultimately be? Right? Is the question clear? You have two, let's say out of all those tested variants, we find two that are significant. One, where the T is a height increasing allele with effect size of 1.1. The second, where T is the trait decreasing allele with effect size of minus 1.1, because having a T reduces your height. And my genotype at this position is DT. My genotype at this position is also DT. What's my height? What's my genetically predicted height, rather? It's 160. Why my, so okay, my height is 160 plus 2.2 because I have two copies of this allele. And I also have two copies of this allele, so it will be minus 2.2. So I would be back to 160. Yeah? So it's additive, where I add my risks or my effects from each independently significantly associated variant. Yeah? Yeah. 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 Yeah, but it doesn't matter, right? It doesn't matter as long as you're consistent, right? As long as you're consistent in about that A as a reference, then you know that beta is the effect of T. You could have done the opposite, where you could have said, okay, I'm going to keep T as a reference, and then the beta would have been the beta of A, which would have been minus 1.1, right? Like it doesn't matter. It would just flip the direction, but the model itself would not change. Yeah. Beta, so you get one beta for each SNP, right? So let's say, okay, why do we need beta? Beta is to know what the effect of the allele is, correct? With respect to the other allele. With respect to allele A, allele T has a beta of 1.1. With respect to allele T, allele A has a beta of minus 1.1. So it's a relative term. The plus or minus is relative with respect to the reference allele. And you can set that, it doesn't matter which one you pick. We pick that according to the reference genome. Right? The reference allele is the one that's in the reference genome. Okay? Yeah. I'll come to you, yeah. Yeah. What do you mean that is different? Yeah. Correct. I see. You're going to set beta naught to be the same for, let's say, all the average height of all the people with the reference allele, with the non-risk allele, right? So that's the beta naught. You're setting the beta naught to be the same across all, right? Yeah, that's a good question. Yeah. 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 You're not, but so what you're actually kind of approximating beta naught with the average height of all the people who have all the reference alleles, right? So that's your beta naught, your average. You're approximating it. You're right. When you estimate the model, beta naught is going to be slightly different, but you're approximating it by the average reference height. Yeah, you're right. Yeah. This may not be the same. This may be 1.5 or 1 point, whatever. Yeah. I don't follow what you, are you saying that the number of samples in each group, will that affect the, I don't understand your question.
I'm saying because of these different groups, that is not the other person. The slope is different because there are different elements for different alleles. Not alleles, I mean different.
Yeah, so that's what 1.1 here was, I just put a number, right? This can be 2 or this can be 3. I don't follow.
So I think
Yeah. Yeah.
there's a
Okay.
photo.
Okay.
I'm sure you have lots of questions.
Yes, and I'm only halfway done, so maybe we can continue this tomorrow. Or come find me. We can, please come ask me questions so I can address all of them tomorrow. Yeah? Okay. Okay. Yes.